In this video lecture, I'm going to talk about complement in health and disease, evolution of rheumatoid arthritis. And as you know that the complement system is a major effector of the humoral branch of the immune system, and it includes more than 30 soluble and cell-bound proteins. This start from C1, as can be seen here. This is C1. It is formed of C1q. It has one main stick and three small sticks to the right, three small sticks to the left. Each small stick ends with a knob. And here we find a body with enzymatic activity known as C1RS. Then this is C4. It has a short fragment C4A and big fragment C4B. Then C2, also it has C2A, which is a short fragment, and C2B, which is a big fragment. Then C3, also it has two fragments, C3A short fragment and C3B big fragment. And then C5, it has also two fragments, C5A and C5B, which is big fragment, up to C9, which looks like a rock. And after initial activation, usually these complement components, they circulate in inactive form. So in the circulation, these proteins, they are not active. But however, after initial activation, usually by formation of immune complex, so the complement or the classical pathway of the complement system get activated following formation of immune complex. And the immune complex is formed of binding this is two antibodies, for example, of IgG. They capture here an antigen, and this we call it immune complex. And this immune complex activates C1. And as can be seen here, this C1q, so they bind to C1q, and this it activates C4, and the activated C4 cleaved into C4b and C4a, and then C2 get activated, and also it cleaved into C2a and C2b. So C2a will bind C4b, and this is C4b. C2a, it is known as C3 convertase. It has an enzymatic activity, and it acts on C3. It cleaves C3 into C3a and C3b. C3b will bind to C4b and C2a, and C4b, C2a, C3b, it is known as C5 convertase, and it acts on C5, and it cleaves C5 into C5a and C5b. C5b will bind to this molecule. Following that, we don't need to cleave more, or no need to cleave more complement components, as the machinery now is on. So C6, C7, C8, they bind without being cleaved. And as can be seen here, from C4b or from here up to CS, they look like a key. So they bind C9, and C9, it looks like a lock. And this molecule, it is known as membrane attack complex. The membrane attack complex has the capacity to remove phospholipids from the surface of the membrane of the cell. So, following this activation of the complement components interacted in a highly regulated cascade to carry out a number of basic functions, including lysis of cells, bacteria, and viruses. As I mentioned, that the complement system has the capacity to lyse or induce lysis by means of this membrane attack complex. So this membrane attack complex can remove, or it removes phospholipids and lead to lysis or rupture of cells and bacteria and viruses. And also complement maybe C3b, it binds to some receptors on the surface of cells, and actually this binding triggers inflammation, and also immune. One of the functions of the complement system is clearance of or removal of immune complexes from the circulation. Excessive immune complexes in the circulation is dangerous in some instances, as if they resided in any organs, they may initiate an inflammatory process. However, the complement system clears these immune complexes and deposit them in spleen and the liver. The spleen and the liver are the gravity of the body. So, deficiency of the receptors on surface of some cells, or deficiency of complement component, may lead to failure of this clearance of immune complex and may lead to autoimmune disorder. For example, we know that African ladies, they are deficient in complement component number 6, and they get usually what is so-called systemic lupus erythematosus. And also deficiency in the receptor on the surface of the body also may predispose to the SLE. So actually I focus on rheumatoid arthritis, but however, as can be seen here, there are 3 fragments, C4A, C3A, C5A, and these known as anaphylaxis, and they can lead to inflammation following formation of the immune complex or deposition of immune complex in some surfaces. For example, in case of post-streptococcal glomerulonephritis, antigens of streptococci, actually dead antigens of streptococci, they circulate and they find their way to the glomeruli of the kidney, and they sit or settle on the glomerular membrane. Nobody knows why these antigens actually they like to go and sit on glomerular membrane. But however, when the circulating specific antibodies arrive to this area, the specific antibodies will bind to antigens of the streptococci, and this will lead to formation of immune complex, which may lead to inflammation or activation of the classical pathway, which in turn lead to post-streptococcal glomerulonephritis following infection with streptococci. But however, this is a sterile infection. Also, in case of rheumatic heart fever, the antibodies against streptococci may cross-react with a cardiac muscle, and this is actually it might lead to cardiac failure, heart failure, or carditis, and the disease is known as rheumatic heart fever and or rheumatic heart disease. So, however, in normal condition, complement component do not attack body cells. For example, due to presence of some regulatory proteins. These regulatory proteins interfere and prevents destruction of or attacking of normal body cells by complement component, and for example, complement 1 inhibitory factor or C1 esterase. And as you know that deficiency of C1 esterase or C1 inhibitory factor may predispose to a disease known as angioedema. In angioedema, the patient present with swelling of tongue, maybe lower lip, swelling of the throat, and this is life-threatening condition. And so the treatment here, this as can be seen here, this is C1 inhibitory factor, and actually when the immune response is done with a complement, so as a feedback, C1 inhibitory factor will get released and inhibit C1 to stop more cleavage of the complement component and to stop more formation of these toxins. As C4A, C3A, these are short fragments of complement components. They lead to release of histamine. So as you know that histamine induces a vasodilatation and increases permeability. And C5, as you know that, stimulate influx of cells. So these short fragments actually increase capillary permeability, causes increased capillary permeability, vasodilatation, influx of cells, and this will lead to inflammation. So when the reaction is done and when the immune responses are done, so there will be inhibitory factors, C1 inhibitory factor, and this will stop and interfere with C1q and in turn stop more inflammation. So in case of rheumatoid arthritis, as can be seen here, this is small joints, hand small joints, and this is case of rheumatoid arthritis. There is hyperplasia of the synovial membrane with synovitis, destruction of bones, and degraded cartilage, and as can be seen here, swelling. So activation of the complement is taking place here in this small area, and actually these anaphylotoxins C4A, C3A, C5A will lead to inflammation, and they will lead. But what is the source? Actually, one can ask, what is the antigen here, and what cells that can reach here and interact? So as I mentioned before, that in case of rheumatoid arthritis, there is an inflammation in the joint, and it is actually, there should be a trigger in case of rheumatoid arthritis. The trigger that predisposes the inflammation in the joint may be infection, may be inflammation, may be trauma. So now the condition becomes abnormal. So under abnormal condition, the cells, for example, T helper 1, T helper 2, macrophages, and B cell, they can enter the joint. However, in normal condition, it is strictly forbidden for cells to come and settle and sit here or enter the joint. But under abnormal condition, following, for example, trauma or severe infections, so cells, they come and sit here. Normally, these immune cells, they die by apoptosis, and in a normal condition, apoptotic cell, when they die, these cells, they are removed by a special subset of macrophages. But however, when these cells enter the joint, and when they die by apoptosis, the apoptotic cell and necrotic cell, they cannot be removed by that subset of macrophages due to absence of that subset of the macrophages. And during apoptosis, the formation of some proteins known as citrullinated proteins. So during apoptosis, citrullation of some proteins is actually a physiological process, and this citrullation, it means that we remove a molecule of arginine, so the protein, it becomes negatively charged. And so citrullation of proteins, this is a physiological process, takes place during apoptosis or during death of these cells. So when these cells come and sit here and accumulate it here, and due to absence of that subset of cells that have the capacity to clear or to engulf them, citrullinated proteins will get accumulated in the joint. Actually, these antigens or citrullinated proteins will be taken by the antigen presenting cell macrophage, and then get processed enzymes in, and then at the endoplasmic reticulum, they will bind to major histocompatibility class 2, and then the citrullinated peptides get expressed on the surface of the macrophage in association with major histocompatibility class 2. And as can be seen here now, T helper 1 will interfere because, as you know, that T helper 1 is a blind cell. T helper 1 cell has no capacity to see tertiary protein, but they can see processed or primary protein. So T helper 1 will interact with presented peptide by the macrophage. This is citrullinated peptide, then get activated. When T helper 1 get activated, they produce some cytokines, for example, interleukin number 2 and interferon gamma. Interferon gamma recruit more macrophages, and the recruited macrophages produce some cytokines, tumor, for example, tumor necrosis factor alpha and interleukin number 1. Actually, this story, it takes place here in this small joint. So cytokines by the macrophage, tumor necrosis factor alpha and interleukin, once they are pro-inflammatory cytokine, and they have the capacity to induce destruction in this area and inflammation. And as I mentioned, that T helper 1 produces another cytokine known as interleukin number 2. Interleukin number 2 activates B cell and T helper 2, so they enter now the game. And actually, the B cell here inside or in the joint, as you know, that B cell is an antigen presenting cell, and also it has a capacity to take citrullinated peptides. So, but however, B cell, they talk only, or they like, there is a crosstalk only, they talk only to T helper 2. And as can be seen here, macrophages, they have crosstalk with T helper 1. So B cell now interact or process the antigen and present it in association with major histocompatibility class 2 to the T helper number 2. And this get to activation of T helper number 2, and activated T helper number 2 produce some cytokines, for example, interleukin number 4, which is a growth factor for B cell, and so B cell get activated and differentiate into B cell memory cell and plasma cell. So plasma cells, they produce antibodies to citrullinated peptides. These antibodies are known as anti-CCB antibodies. So when anti-CCB antibodies encounter citrullinated proteins here due to failure of clearance of apoptotic and necrotic cells, so this will lead to formation of immune complex. And here, as can be seen here, the immune complex is formed from anti-CCB or anticyclic citrullinated peptide antibodies. So when they encounter these citrullinated proteins, they bind to it, and this will lead to activation of the immune complex. This will lead to formation of the immune complex, and the formation of immune complex will lead to activation of the complement component, and this activation actually starts by activating complement component number 1. So this following that complement component number 4 and 2 get activated, and then complement component number 3 get activated, and then complement component number 5 up to complement component number 9. And as can be seen here, the activation of the complement component is not in order. For example, here this is C1 get activated, complement, then C4, 2, 3. 3 comes after 4 and 2, and this is actually it is due to discovery of these molecules. So here this is a story of the evolution of the rheumatoid arthritis due to activation of the complement by peptides, by proteins known as citrullinated peptides. And as I mentioned, that citrullinatation is a normal physiological process during apoptosis, cell apoptosis, and actually the citrullinatation takes place by removal a molecule of arginine, and this will lead to negatively charged proteins. But however, as the citrullation takes place here inside this area and this inflamed area, and due to failure of the clearance of the citrullinated proteins and the apoptotic cells and the necrotic cells, these cells get interacted here, and this will result in formation of antibodies to citrullinated peptides, and these are known as anti-cyclic citrullinated peptides, antibodies. However, anti-citrullinated or anticyclic citrullinated peptides, these antibodies, so the test or detection of this could be in early stage of rheumatoid arthritis. So they have, these antibodies, they have predictive value even before appearance of symptoms. And also these antibodies, I mean anti-CCP antibodies, they have diagnostic value, and also it has a prognostic value. So you can follow the efficacy of the treatment by detecting these antibodies every then and now. And also it has been mentioned that some individuals may produce antibodies of IgM against their IgE. So here's IgE, it acts on antigen, while this IgM is acts on antibody. So when they interact, this will lead to formation of immune complex, and immune complex will activate C1, then C4, 2, 5, up to C9, and this will lead to formation also of anaphylotoxins, and these anaphylotoxins will lead to inflammation here, and this will lead to rheumatoid arthritis, as can be seen here, characterized by hyperplasia of the membrane, destruction of the bone, synovitis, maybe tendinitis, and destruction together with swelling and hyperplasia of the synovial membrane. Thank you very much, Professor Ahmed Moulad.